So, which is translation exploitation. And we've been hearing from Geoff, Scott, and Paul so far, I think, in this discussion about the environment. So, but if I can ask them to take it away for the next 20 minutes or so to present their thoughts in this area. Thank you.
Okay. All right. Hello again. Yeah. So this section of the Grand Challenge will involve the exploitation and translation of research and discoveries stemming from Euglena and Euglenids. So on the panel today, we have myself, Scott Farrow, Geoff Horst from Kemin Industries, and unfortunately, Paul, who you heard from this morning, couldn't attend this afternoon. Paul did ask that we just briefly have a moment of silence for COVID-affected individuals. So I propose that we perhaps take 30 seconds to do that, if that's okay with everyone.
Please go ahead. Go ahead, please.
Okay, thank you very much. Okay, so I thought I'd begin kind of this session and just kind of outlining some of the major global challenges that we're facing today. Although we live in a very great world where, you know, a lot of us are the recipients of a lot of the benefits of scientific discoveries over the last 50 years especially, there's still many areas in which we need solutions and creative solutions to solving these challenges. And, you know, we have things like energy, science, technologies, climate change. And this slide is just to focus on and present to you the different areas in which, you know, technology could help make improvements. When I first started working on Euglena, you know, I didn't realize all the different potential that these small eukaryotic microbes had, but they definitely do have a wide-reaching range of applications that they can help combat a lot of these different areas. So there are hundreds of euglenids, and traditionally, I guess, we've focused really on Euglena gracilis because it's quite easy to grow and it's robust. But if you can think of all the different euglenids that are present in the environment, each one of them is a potential treasure trove of genetic parts. And with the advent of things like synthetic biology, as mentioned before, and combinatorial biochemistry, and even biocatalysis, there are plenty of enzymes and probably a lot of very, very unique enzymes that could be leveraged for these emerging fields. So I think that, you know, one of the challenges that has already been mentioned is, you know, we need more sequencing. We need more sequencing and we need more functional annotation of these genomes. And that is going to require things like reverse genetics tools, et cetera, to help us interrogate the function of these genes. So I'll just reiterate, you know, there's a lot of potential within that genetic information for discoveries that can help combat some of these challenges. We've seen recently the kind of directed evolution of plastic degrading enzymes. There's lots of promising things that Euglena holds within the genetic space that we could perhaps leverage to combat these problems. Similarly, if you think of the genetic parts, you also have all the different products of these plethora of biosynthetic pathways, even secondary metabolites. And I'd suggest that bioprospecting for new metabolites with new biological activities and screening them, using classical kind of activity assays would be one thing that we could do as a community to help unearth the new functionalities of some of these chemistries that are found across these different species. I would say that, we know some of the industries that euglenans are being used in now, food, environmental remediation, energy, and materials, but I really enjoyed the comments about using it as, thinking outside the box. Can we use this as a microbot? That's a cool idea. So fostering, as a PI, or if we can collaborate as a company, really kind of outside the box thinking to, address some of these global challenges. So I would encourage everybody to think outside the box and try to come up with solutions and technologies that could help. I had a comment here from Paul that he wanted me to include, and that was, some of their preliminary work with Euglena ficin as an adjuvant and a treatment for to prevent angiogenesis and mTOR activation suggests its utility in anti-cancer therapy. So he suggests that parallel testing for antibacteria, fungi, virus activity should be completed as well. Okay, so that's kind of the slide wrapping up the discovery and innovation. I'll also just like to bring to your attention that, with the commercialization process, for food especially, it has to go through a very rigorous, can go through a very rigorous regulatory process. And we recently went through this as a company, and I know that Geoff commented about that earlier as well. And this can cost hundreds of thousands of dollars to do toxicology studies, and a large reason for that is, there's a lack of widely available data on human consumption or use. So you have to go through this process and, do toxicology studies. You have to demonstrate a consistent production methodology with in-depth descriptions. You have to have very stringent biomass profiles, similar protein, amino acid, carbohydrate, fat, etc. And it's quite a back and forth GRAS panel application with the FDA. And the whole process can take around 1 to 2 years. So just to provide a little bit of clarity on, once you get the discovery, there's still a lot of work, and that's where partnering with an industry partner can be quite helpful. Okay, the other thing is, when we look at Euglena, we think, okay, it has all these different growth modalities. It can grow in the light, it can grow mixotrophically and in the dark. But I think one of the requirements to help commercialize Euglena in the food space, etc., is coming up with new fermentation processes that perhaps aren't as traditional as you might think. So currently, we grow these things in large bioreactors or in ponds, but are there other ways that we can grow Euglena to access new products? Can we leverage some of the information from the past? Can we look to the past to see, find new opportunities or continue new opportunities with having the benefit of kind of a modern scientific approach? Can we use the knowledge of Euglena behavior to create new novel growth strategies? There's also quite a requirement now, to come up with maintenance and genetic stability strategies for long-term growth and reproducibility of a certain Euglena strain. Obviously, contamination, we don't have to use antibiotics when we're growing Euglena, but with that comes another suite of problems is, we can often get a lot of contamination. So we need to overcome that challenge, and it could be an engineering effort, as I suggested in the past with talks with Thank God. It would be good to get some engineers involved to think about well, how can we build something that can do better with contamination with an unconventional kind of microbe? And that's all I have for now. I'll pass it over to Geoff.
Thanks, Scott. Hello, everyone. So maybe if you had listened in yesterday, you heard a little bit about my background, but for those who are joining for the first time today, I'll give a quick background on myself. So I started a company called Algal Scientific back almost 10 years ago now, and we commercialized the first beta-glucan products for animals and for humans. And so we were taking advantage of all the prior research that had been done on beta-glucans and the way that they can modulate the immune system of animals, including humans. And about 3 years ago, we were acquired by Kemin. And so Kemin now continues with the commercialization. And so we have our animal products pretty much in every major country. And if you had chicken or pork or fish recently, there's a decent chance that those animals were fed with products that came from Euglena. So we're already having an impact on the food supply. And then also recently, we also launched products for plant health. So you might not know, but plants also have an immune system that is responsive to beta-glucans. And so it's early stages, but we have some products that are now in the agricultural space. But what I wanted to share here was more of the kind of commercialization business side of how do you get products into the market and what's required there. And so really the first part is understanding what the market wants and how much it's willing to pay for it. So I'm going to use the algae biofuels story as kind of a template for this, because I think probably a lot of us were familiar with that. The problem with algae biofuels is that the research groups, including companies and venture capital, they way underestimated the cost of making the fuels. And at the same time, they way overestimated how much customers were willing to pay. And so that's why you don't find algae biofuels, even though we spent billions of dollars, including public taxpayer money, to try to do that. So I hope that Kengo's team is able to break through that barrier, because I personally want to be driving cars with Euglena biofuels. But up until now, it has not been successful. And so kind of using that as a template, if you're trying to develop a Euglena-based product, what you should be asking yourself is, what is that product going to do for the customer? And so I like to use the kind of the description, what job is it that this product is going to do? And so from that algae biofuels one, the job isn't just to provide a liquid fuel, right? The goal there was also making the customer feel like they're also saving the environment or not having nearly as bad of an impact because they're not burning fossil fuels. So it can be a complex job that you're trying to do, but that's where you kind of need to start from. What does the customer want? And then you need to figure out, is this kind of a nice to have product or a need to have product? If you're developing a vaccine, that's a need to have product, right? If you're developing a source of vitamin E, that's a nice to have, or vitamin C, but there's other ways to be getting that same ingredient. And so that's going to dictate then how much is your customer going to be willing to pay for that product. And so if you have a really niche kind of offering, so let's use the biofuels one as an example, maybe somebody's willing to pay twice the amount of for a Euglena biodiesel than they would for regular diesel, right? But that's not going to, not everybody's going to be willing to pay twice as much, right? But there will be a market for that. And so that's kind of establishing what is the market willing to pay for you, for your product. So Sky, if you could go to the next slide. Is there one more between those?
There you go.
Yep. So now on the cost side, this is where the biggest error probably occurred with the algae biofuels model. It would have been a simple desktop exercise for all those venture capital groups to actually figure out what is the real, when it comes down to the nuts and bolts of it, how much is this going to cost us to make an algae biodiesel? And they could have figured out and saved billions of dollars if they just figured out that it takes a whole lot of energy, which equals money, to be able to concentrate algae into a paste and then do the extraction of those lipids. And so just a very simple exercise would have told them, this really isn't going to make sense to make biofuels for less than, let's say, $2 per liter. And if you can't get it less than $2 per liter, it's really not a very effective biofuel. So for your product, hopefully you're going after much more valuable, products. But my suggestion is that after you've kind of demonstrated that you can make this molecule, the target molecule, at a scale of, let's say, 1 liter or a 10-liter fermenter or an open pond, that's where you really have to go out and get an independent group to really do your product. Because that's gonna, like, if you can't show that you can have your unit economics be low enough to get a certain price point, then you really need to take a hard look to figure out, is this something worth continuing, at least at this point? Maybe you still need a complementary technology to make it happen. But instead of wasting years of your time and venture money or taxpayer money with grants, you really need to figure out, am I even close to getting to the right economics? And that's where a lot of companies fail, and that's okay. We need to have lots of failures to have a couple wins, but before you get too far along, you got to start bringing in people that can figure out the cost model. And part of those groups could actually be a contract manufacturer. So that's what we looked at for Algo Scientific and for Kemin. Once we figured out how to grow Euglena, let's say at the 20-liter scale and the 500-liter scale, then we went out and started talking to contract manufacturers to figure out, okay, if we want to make 5 metric tons of this material, how much is it going to cost to go to a contract manufacturer? Or if we say that we want to do 500 metric tons per year, what's the price point? And certainly the bigger scale you get, the lower the price point per kilogram of your biomass. And then the last part is really, what is your go-to-market strategy? A lot of us that are participating today are very early in the development and R&D stages, and so we probably don't know that much about how do you actually get products to the end consumer. And so that's where you're going to need to have an industry partner probably to be able to finally sell this product. And so Scott, if you go to the next slide. So this is kind of where I've mapped out this continuum between the discovery of a really cool new molecule all the way up through the development of that. So that's where you kind of scale up the production. That's where you start doing some of your efficacy trials, let's say. And then you have people that can actually make this biomass at a very large scale. And then you finally have the group that's actually going to be selling to the customers. And so, no company is going to be perfect at doing all those things. Perhaps in the olden days, pharma companies did all the research all the way up through distributing it. But more and more, even R&D companies are really only acquiring technologies after they've done the basic research. And so Pfizer and a lot of the big pharma companies, they don't even do that much R&D any longer. They actually will just acquire the R&D from a development company, and then they take it to market. And so as you see yourself on this spectrum, there are some limitations, and you should kind of be aware of those. So kudos to all those people that are in the kind of the basic R&D stages. That's where we're going to find some really interesting golden nuggets. But if you're in academics, this is generalizing here, there's not a whole lot of incentive to actually get that to market, at least historically. So universities weren't built upon trying to get those products to market. That's changing a little bit, and you can kind of look at MIT as one of those universities that really prioritizes getting R&D out into the commercial marketplace. The problem with some of the big companies that are facing the customers is that they understand what the customers want. However, they're usually not very innovative. They might not have a very good R&D department, and so they're going to be pretty slow to respond. And in fact, they're sometimes the hardest customers to convince that you have a product that customers might want, because they're going to say first, like, I don't think that they're going to want that. And so that's where these kind of middle stage companies, they're the ones that you find with startups, because they actually see a future that the end customers don't even see for themselves yet. And so that's where Algo Scientific participated in this stage where we didn't invent the research about beta glucans. So we really didn't do any of the R&D on beta glucans. What we did was figure out how are people growing it, and then we figured out how to grow it at massive scale and then take it to market. So no company can be perfect at everything. And so my suggestion, and kind of echoing what Scott just mentioned, you should probably look to companies that can kind of get you closer to that endpoint of the, how do you actually get it to scale and how do you get it to the marketplace? So my suggestion is don't try to do it all on your own. And there's a lot of opportunity for everybody to, if you're selfishly and you're venture backed, everybody needs to make money, but that's how the real world works. And so it all comes down to money to some extent. And so somebody's got to make money. So happy to take some questions after this.
Thanks very much, Geoff. I think that was a very insightful set of comments, and I think that we up in Dundee have a sort of mini pharmaceutical company embedded within our biochemistry division, and certainly the slogan up there is to fail early. That it's quite important to be realistic about the probability of success of a particular drug in this case, and that if there are some red flags, it's really quite important to make sure that you don't use up resources on something that's ultimately going to collapse, several years and several $1 million down the line. Okay, could we take some questions then? Where are we? Sorry, I'm just trying to... So I've got a comment from Alistair Simpson for the impractical amongst us. When considering biomass, food supplements, etc., what are the main 1 to 2 comparative advantages of Euglena when compared to other microalgae? A very good question indeed.
Scott, you want to field one of those and I can add anything?
Sure. Yeah, I think that Euglena has a unique kind of protein composition. It's high in protein. I would, as an analogy, you could think of it of having multiple levers to kind of play with. So we know that it makes healthy fats. We know that it makes proteins. We know that it makes glycopolymers. So as a solution, it's got a lot of different natural products that it's making compared to some microbes, and we don't even know the whole range of natural products that these things are making. So there's a lot of potential there, I think.
Yeah, I think one that we take advantage of a lot is the fact that it doesn't have a tough cell wall. And so if you're making a product that is produced internally, like paramyelin for us, but pretty much any of the molecules that we're talking about here, the fact that you don't have to crack that cell wall, at least if you're trying to extract it, or even if you were to have an enriched whole cell algae, the fact that if you feed whole dried algae, Euglena, to an animal, including humans, you don't have to do any extraction because that protein skin, that pellicle, dissolves in the stomach. And so that saves us a whole lot of money and effort, and it allows you to have a very clean label product because you don't have to do any extraction. So other algae, even like chlorella or yeast, of course, you have to do expensive extraction. And so we have the advantage of having a very thin and digestible protein skin.
Okay. I have a question or a comment from Kevin. He's asking about the carbon footprint of agricultural feed, and particularly for aquaculture, which he notes is considerable. So is Euglena potentially a better solution? Does it have a lower carbon footprint than some of the other solutions that have been used for agricultural feedstuffs?
I can make a brief comment about that. It depends on a lot of things. It depends on where you're manufacturing your product, where you're selling your product. If you can, get over those hurdles, if you can come up with a kind of distributed manufacturing approach, that can be very helpful in combating, your carbon footprint. So that's one thing that people can look at. It depends on how you grow Euglena too. Like we know that you can grow it heterotrophically and, you know, phototrophically. Clearly, phototrophically, you can capture carbon that way. Heterotrophically still relies on the use of, you know, sugar inputs. So I think it depends. It depends on the strategy. I don't know, Geoff, if
Yeah, there's a lot of interest right now in the aquaculture world about using algae for as sources of protein because we're trying to get away from fish meal. And so fish meal is not a very sustainable source of protein for fish feeds. The challenge really comes down to cost again. So yes, you can make Euglena, feed it to fish, feed it to zooplankton, and it's really readily digestible. It's a great fish feed. The only challenge is the cost, right? And so fish meal is about $2 a kilogram, and it's a perfect source of protein and lipids like DHA. But it's very difficult to make Euglena for $2 a kilogram. And so I don't see it ever becoming a major source of the diet. I think it's going to have to be something that's very specialized. Like, are we going to be able to enrich it with some sort of vitamin or some sort of other value add besides just the protein? Because the protein and the fats alone are only worth $2 a kilogram. And it costs a lot more than that to make Euglena, no matter if you're photosynthetic or heterotrophic.
Yeah, absolutely.
Okay. Sorry, Mark, I have a question. With regards to, thanks Geoff and Scott for the presentation. With regards to the resources to maybe actually cultivate Euglena for nutritional purposes versus cultivating for biofuel, do you envisage any competition in terms of resources? Should one rather, you know, just as you're mentioning, focus on, you know, trying to feed the world first, and then trying to how would that resources trying to do both? How would they compete with each other?
Okay, we have a question from Sergio Guerrero. He's interested in if grassless is being used for beta-glucan-induced trained immunity. I'm not totally sure what he's driving at. But,
I think I know what Sergio is going after here.
yeah.
That's exactly what Kemin's products are doing. So we're using Euglena for the paramyelin, and so we're giving very specific doses of beta-glucan to animals and humans. And so the nice thing is that you don't need a whole lot, right? And so for humans, we're giving them about 350 milligrams a day. That gives you basically the right dose of beta-glucan to do that immunity training. And for animals, it's like about 150 grams per ton of feed. So that's what our products are focusing on, is the beta-glucan immunity strategy.
Okay, thanks very much, Geoff. And I think probably one more question that we have here is from Ainsley Lewis, who is interested in a link to other industries who want to give away free waste. So, for example, the dairy and alcohol industries. Or could you grow them in sterilized sewage water? I guess he's looking at or thinking about ways for culture cost minimization.
Yeah, I think that fits in nicely with what I mentioned previously, is coming up with novel strategies to grow Euglena. Certainly, if you can find somebody willing to give away their stuff for free, then it's definitely something that can be explored. For sure. And it is a potential way to cut costs dramatically.
Absolutely. Okay, I think we should probably call the session to a close, unless there is somebody with a massive burning question. No? Okay, so